Hello students, this is Dr. Faizan Mirza. Here I'm discussing with you the October-November 2025 variant second of paper 2. Now in this paper, you have the first question that deals with villi. They have told you that the wall of intestine is shown to you, it is highly folded to form villi and villi is shown to you, as well as there are sections shown where they say that a single villus is shown and a crypt of Liber Cohen is mentioned. And there are in these are the enfoldings present between the villi. So you are told what Liber Cohen or the crypts of Liber Cohen looks like. So this feature here shows to you the structure of villi. On the surface of these villi, you have inside you have blood vessels. It's like in the center you have a lymphatic vessel. The cell, this is told to you that this is a capillary network. The cells here are called enterocyte. Then there's a goblet cell as well. You can try to recall what the goblet cell is. And outside you have the gut lumen, which means the food, the soluble food digested material will be absorbed in this direction. Enteroendocrine cell is also shown, and you can see these the small spots are mentioned are shown here, which means that it's an endocrine cell, so it must be secreting something. Then in the crypt of Liber Cohen, if you see that this region is the crypt of Liber Cohen, in the crypt of Liber Cohen, you have the Paneth cell. This is mentioned, this is the Paneth cell, and then there's intestinal stem cells. So just by looking at the diagram, and then they have shown to you a venule and a lymphatic vessel. And in this way, they have asked you that this thing here, X is also a vessel. So you can see the color tone of this diagram that this vessel X and this entire capillary network is having the same kind of shade in the diagram, whereas the one which is in the venule is the same as here. So we know that in venule you have deoxygenated blood. So this part of capillary network is deoxygenated blood, and this part of capillary network is the oxygenated blood. So the capillary is receiving blood supply in this direction. So if it's receiving blood supply in this direction, which means that the vessel X is a vessel just before the capillary network. So this must be an arteriole, because we know artery divides to form arteriole, and an arteriole joins to like eventually divides to form capillaries. And then capillaries join back to form venules, and the venules then join back to form veins. You know that path. Okay, so this figure shows you how this goblet cells and the enterocytes are shown. The apical surface is mentioned. Okay, that's now the move on. Now let's move on to the questions. A student incorrectly stated that enterocyte has many cilia on its apical surface. Explain the difference between cilium and the microvilli. So cilia is for increasing the surface area. No, it's not for increasing the surface area. It's for moving the mucus across, like over the surface. Plus, the cilia and flagella in eukaryotes, they have a 9+2 arrangement with the basal body of 9 triplets of microtubule. We studied that in cell structure. Whereas the microvilli increases the surface area for reabsorption and secretion, it is way much shorter than cilia. So just one role of the mucus produced by goblet cells in the villus. So the mucus reproduce, it will provide a layer. It will prevent the cells from damage from the enzymes against the entry of pathogen, as well as they will reduce the friction and allow the food contents to move easily along. They have asked you which vessel is this. Already I've discussed this is vessel X is arteriole. Describe the formation of tissue fluids. How the tissue fluid forms, we know at the arteriole and the capillary, at the capillary, the blood hydrostatic pressure is high. This allow pressure-driven filtration, means that your blood filters out, plasma moves out, and it moves out through the endothelial pores or the gaps in the capillary wall. And plasma leaves and enter the tissue space and become the tissue fluid. This tissue fluid is rich in glucose, amino acids, ions, and small solutes, but they lack the large proteins. The large proteins, they remain in blood. Next, they have told you in this question that there are certain cell signaling molecules such as GLP-1. This initiates a number of responses in different body cells. Some of these responses include increase the release of hormone of insulin from pancreas, decrease the acid from stomach. So two organs are being affected by one signaling molecule. So this one signaling molecule can identify pancreas as well as the stomach. So this signaling molecule must be able to reach both. So we know the cell signaling molecule reach their target cell through blood. Outlined in sequence, the main stages involved in process. So the command word is outlined, which means your answer needs to be in brevity. So we say we just describe this in a very brief way, just moving around the outlines that GLP-1 enters blood, it reaches the target cells to reach stomach and pancreas because two or different organs are identifiable for this particular signaling molecule, and the signal molecule then binds to the complementary receptor. The receptor must be complementary in shape for it to attach. This triggers a series of events inside the cell leading to response via signal transduction. Then Paneth cells are shown to you. This is the Paneth cell. The Paneth cell is shown to be having these large, you can see rounded structures, and the nucleus is there. Further, they are saying Paneth cells are formed by mitotic cell division. In sequence, they're asking you to list down the stages of mitosis. So it's prophase, metaphase, anaphase, telophase in the order. You get 1 mark for that. Lastly, in this, towards the end, they're asking you that in this figure 1.2, state and explain the evidence that yes, the cell is actually a secretory cell and it can synthesize peptide and proteins. How do you know that? There's a lot of Golgi. You can see a lot of vesicles fusing with the cell membrane. And the rough ER is there. The abundant protein synthesis of mitochondria is available for providing ATP for the movement of vesicles towards the cell surface membrane. And lastly, they're asking you explain why Paneth cell has a very different appearance than the intestinal stem cell, because the intestinal stem cell is not a differentiated cell. It's not a specialized cell, whereas Paneth cell is a differentiated specialized cell. If we go back to the diagram, let's see what do you see here. All this is the mitochondria. This mitochondria provides energy. This is the mitochondria. This is the mitochondria. All these are mitochondria. And these circular structures are the vesicles that can undergo exocytosis. Plus these membrane-bound tubular sacs. These are all the cisternae of the rough ER. So you have a lot of these structures that show that this particular cell is involved in secretion and synthesis of proteins. Next, they are asking you about cholera. A typical question is when they ask you to name the species of a bacteria that cause cholera. So whenever they ask you the name, so you have to be very specific and it must be spelled appropriately, because even if an O or an E or an I goes missing or you misspell it, you lose the mark. So just explain how these two approaches prevention of control of cholera. For example, how safe drinking and personal hygiene. So contaminated water should not be taken for drinking, for cooking. Transmission cycle continues. Otherwise, the disease transmitted by fecal oral route. Water taken from underground sources must not be used for drinking unless it is boiled or chlorinated or treated with UV. Why? To kill pathogen. You have to explain. So whenever you are addressing the question for command word explain, you have to give a reason. So yes, to kill pathogens. And water pipe should be directly connected to water treatment plant to avoid contamination. People should avoid drinking directly from open sources such as river, lake, and ponds, because they are asking you for personal hygiene as well. Like at the personal level, what a person can do apart from just what resources he or she is having access to. Furthermore, they are saying in this next part that mass vaccination is possible using a vaccine OCV in which the disease is like highly spreading. There is a process of passive immunization that involves transferring antibodies. So which antibodies do we transfer? We transfer the monoclonal antibodies, the MABs. We use MABs, we prepare MABs in lab. We get these monoclonal antibodies and we inject the individual with these MABs, and they give instant uh, relief or instant outcome, because these antibodies go and bind to the target sites within the patient's body, because the patient's own immune system will take a very long time, approximately 2 weeks, to make his or her own antibodies, because the clonal selection, clonal expansion have to occur. So passive immunization gives a very quick response, because this is how the immunity gets boosted on a short-term basis. First, they're asking you the component cause of oral cholera vaccine versus passive immunization. So this vaccine, whenever we are injecting a vaccine, it's always an antigen, whereas for passive immunization is always the antibody. Since it's an antigen, this is artificial active, the other one is artificial passive, which stimulates the production of memory cells. When antigens are there, memory cells will produce, yes, it will be for much longer time and duration of immunity will be longer, it will take more time. Whereas when you put antibodies, lymphocytes do not turn into memory cells. This occurs very quickly. but its effects are not very long lasting. Name two types of cells that are used to fuse to form hybridoma cells. So we form hybridoma cells by fusing the plasma cells, which are derived from the B cells, the B lymphocytes. They secrete an antibody, the type of antibody that is having a specific antigen binding site. These plasma cells then cannot divide. We know that they will die in culture, so they do not divide on their own. So we have to fuse them with the myeloma cell to make sure that they keep on reproducing and divide by mitosis. Now the next question here shows to you a gene LCT that codes for an enzyme lactase in babies. The lactase synthesis is necessary for digesting lactose, the sugar found in milk. Then they've mentioned there's another gene, MCM6. It's having introns that have a regulatory role in expression of the gene LCT. The gene MCM6 codes for a protein that has no involvement in lactase synthesis directly. As children get older, M6 introns are responsible for a decrease in lactase synthesis. This decrease in lactase synthesis is known as lactase non-persistence. Okay. LCT and MCM6 are located on different chromosomes, suggest differences between these two genes other than their location in different positions of the same chromosomes. Now, you have to understand that you are at AS level. You don't have to be, you can say, a bit intimidated by the detail that they give, that this is something completely out of the blue that they have given, completely something not in course. They give detail to test your comprehension skills. At the end, what are they asking? They are just asking you how the two genes differ. So we know the two genes will differ on the base number of bases or the different sequences of bases of DNA that they are having, the different proteins that they will form. They will have different number of introns and exons, and hence the different number of bases there in the DNA, and the DNA fragment length will be varying. So this is typically AS question, but with an intro that feels intimidating at times. With reference to the process of lactase synthesis, explain the relationship between a primary transcript and mRNA. So we know in the eukaryote, whenever the DNA template is being turned into RNA, initially we have a primary transcript forming. The primary transcript then is used as a it is complementary to the template strand. This transcript then undergoes modification. We know we remove the introns, we cut the introns, we join the exons, and this exon joining is by the process of exon splicing. So exon splicing occurred, and this produces the mRNA, which is ready for translation by the ribosomes. They're asking you that for this, the lactase persistent, the mutation, the number of nucleotides do not change. If the number of nucleotides are not changing, what kind of mutation should be? This must be a base substitution mutation. For example, the number of nucleotides were 10, for example, 107. Let's just randomly. And still after mutation, they are 107. It means this was base substitution. Now, let's suppose if they say that the number of nucleotide from 107 turned 106. What kind of mutation is this? So this is a -1 mutation, which means this must be deletion mutation. If they had said that no, the number of nucleotides changed from this and became one more, so this must be addition mutation. So insertion mutation. So we have base substitution, base deletion, and base insertion. So when it is +1, which means additional base, this is base addition mutation or base insertion mutation. When a base gets deleted, this is base deletion. Both of them are frame shift mutations. Whereas if the number of bases stay the same, and yes, mutation has occurred, so this must be base substitution mutation. Then they are asking you to draw the formula for alpha glucose. Just look at the number of OHs, and I think that's a cramped thing that you can easily draw. Then the next question comes uh, for colorimeter. They are saying that there is an artificial substrate ONPG, and this solution ONPG is colorless, but when it is hydrolyzed by the enzyme, it releases a colored product. So we know that as the reaction will proceed, the product will increase. When the product will increase, it means there will be more of color in the reaction mixture. So more color in the reaction mixture is shown to you by this particular this set of words here, that the product is colored. So if the enzyme is working very fast, a lot of color will be produced. Let's move on. Explain why the student chose to use a colorimeter. A colorimeter allows detection of color intensity, a fact. ONPG is producing colored product, we can get quantitative results of the numerical values. And the judgment is not by eye, so it's not subjective. It helps us to get precise, accurate readings, and a graph can be plotted, results showing the effect of lactase concentration as well as a calibration curve to compare rates of variation of the product concentration. Later on, they are giving you two examples of percentage activity in which they have given you immobilized lactase and free lactase, and they have shown to you how the immobilized lactase percentage activity stays high even at higher temperature, and at pHs, still at a higher pH, it's more active. So what do? If they are asking you a question of describe, you take a temperature, you code the value of both to say this is higher, this is lower. Similarly, the ending temperature, like this is denatured almost, it's still functioning. So you code the values with the particular reference. Similarly, here you can say this is a short, this is a slower increase, whereas here this is much steeper. And it continues to like even at a lower 5.5 pH, this is showing a higher activity. You will code the value as compared to the other one. Similarly, at the maximum pH of 9, this is showing a lower activity, and the other one immobilized is still showing the higher activity. So this is how, and the optimum thing is not affected. So the idea is that whenever they are asking you, so you just code these values. When they are asking you for describe, they're asking you with reference, explain why the results indicate that this is an advantage to use immobilized enzyme. More yield, more products, immobilized enzyme are more thermostable, and then you just code the values for the relevant pHs and temperatures. Question 5 deals with bacteria. So question 4 deals with bacteria, and they have shown to you a gram-negative bacteria. A gram-negative bacteria, already mentioned, again, they try to intimidate you with information. You don't have to get overwhelmed. And the gram-negative bacteria have a cell wall. We know every bacteria is having a cell wall, but outside of a cell wall, there is a layer known as outer membrane, and this is present in bacteria which are called gram-negative. The gram-negative bacteria is having an outer membrane, so you can expect that there will be the cell membrane. Outside the cell membrane, there will be the cell wall. Outside the cell wall, there will be the outer membrane. So this thing is the cell membrane, the cell surface membrane. This thing is the cell wall, and this thing is the outer membrane. And inside you will have the cytoplasm. So this is how the bacteria will look like. There will be a cell surface membrane and the cell wall, just like anything else. But an additional structure of outer membrane will be present around the bacteria called the gram-negative bacteria. This diagram shows to you the detail. If we look at the detail, this is the cell surface membrane, the cytoplasm. We know the cell surface membrane is made up of the phospholipid bilayer. The cell wall, where do you see the cell wall? So the entire cell wall here is having the outer membrane. So in this, with the cell wall itself is having the outer membrane here. So what do you have here? The cell wall is having two parts, the outer membrane and the periplasm. What is the outer membrane? This part is the outer membrane, and this part is the periplasm, and both of them are outside the cell surface membrane. So in short, there is a cell surface membrane, and outside there is a cell wall, and within the cell wall there is the outer membrane. Now, what do we see? What's in periplasm? In periplasm, there is a peptidoglycan layer, and there are periplasmic proteins. Every bacterial cell is having peptidoglycan cell wall. So this is nothing new here, nothing new. But periplasmic proteins are new. Then outer membrane proteins are new. This is the outer membrane protein that resembles like a channel protein or a transport protein. In the outer membrane, there are phospholipid monolayer, not a bilayer, it's a single layer. And on top of this single layer, there is a structure called lipopolysaccharide. Lipopolysaccharide appears to be a lipid with a polysaccharide structure. And this is then the external environment. So this entire thing is the outer membrane. And this is the cell membrane in between the periplasm. Together, this makes the cell wall, and then this is the cell membrane, and inside you have the cytoplasm. Okay. Here they are asking you, with reference to figure 4.1, outline the similarity and differences between the outer membrane and the cell surface membrane of a eukaryotic cell. So we know the cell surface membrane is made up of the phospholipids. So here both phospholipids are there too. The channel protein is there as well. But the difference there we have in eukaryotes, the cell membrane is in the bilayer, where here the outer membrane is having a single layer. There's no cholesterol here, so there's cholesterol present as well. So these are the things that we can mention. In the next part, they are saying that the bacteria can end up producing two kind of vesicle, OMV and OIMV. Now, what do you see? This is the bacterial cell. Inside the cell, these are the plasmids. You can just look at the key that these rings are shown as the plasmids. The two kind of vesicles which are forming, one of the vesicle is coming from the cytoplasm and becoming OIMV. It is having the plasmid and these gray things as well, which are shown to you the different cell products. Then both of the vesicles are coated by the same things which are present outside the outside the cell wall or in the outer membrane. And the periplasmic proteins are there, these hexagonal structures. So what do we see? Which vesicles have the periplasmic proteins? OMVs having the periplasmic proteins, because OMVs arising from this outer layer of the bacterial cell wall, which means outside the cell surface membrane. Whereas the vesicle forming from within the cytoplasm is coming from inside. So two kind of different vesicles are forming. With reference to figure 4.2 suggests why fewer of these are formed than OMVs. So these ones require more content to be added, two membranes and a lot of cell wall have to be broken. So ATP need the requirement will be more. So just why ATP is found in OIMV and not OMVs, because this is coming from the cytoplasm. So it is bound to have ATP, whereas OMVs are coming from the periplasmic space, so no ATP will be there. So just by explain why the discovery of these OIMVs have implication for antibiotic resistance, because they contain plasmid DNA. Bacteria can pass on this plasmid to others, and this can code for enzyme that can break down the antibiotic or code for membrane protein that can pump the antibiotic out of the bacteria. The last part, question 5, deals with simply a plant diagram, leaf diagram, cross section shown to you as A, B, and C, A, B structures. A, they are saying this is some region A. Above the upper epidermis, this must be the cuticle. B is the spongy mesophyll, and then there's the lower epidermis. They're saying that you need to just give an example of how a particular adaptation works. For structure A, we can have much thicker cuticle in 0, 5. The epidermis will have trichomes, it will be multi-layered. The lower epidermis, we have no stomata on the lower epidermis. So these are just simple facts that you can recall and state from there. The last question deals with pulmonary circulation in mammals. Deoxygenated blood becomes oxygenated. Okay, they are saying that they are asking you, describe the sequence of events in the heart that allow blood returning in pulmonary circulation to enter the systemic circulation. So from the pulmonary circulation, we know the blood gets like oxygenated. During diastole, this valve opens up and blood entering the left atrium from pulmonary vein reach left ventricle. Atrial systole push the remaining blood. So this is how these keeps on moving. So atrial systole will push the remaining blood into ventricle. When ventricle systole occur, the pressure in the ventricle will rise above the pressure of atria and bicuspid valve closes. You just give the basic description of how the blood exits from the heart to enter into the systemic circulation. This thing here shown to you is the hemoglobin molecule. Describe the quaternary structure of a hemoglobin molecule. So what do you see the hemoglobin molecule is having four chains, two identical alpha polypeptide, two identical beta polypeptide. We will use the term identical alpha polypeptide, identical beta polypeptide, and then there will be an iron Fe and Fe plus 2 state for each, which is the prosthetic group and the heme group. A total of 4 heme groups are there. Outline how a hemoglobin molecule can become fully saturated with oxygen to form oxyhemoglobin. So we know that oxygen binding site is at the iron ion of the hemoglobin molecule. A total of 4 oxygen molecules can bind reversibly at each of the heme group, forming oxyhemoglobin, which is HbO8. The binding of the first two changes the shape of hemoglobin, which means cooperative binding. So cooperative binding leads to every oxygen molecule getting attached to the hemoglobin molecule with much more ease as the previous one. And that's how you settle for this whole paper. I hope this video was helpful for you. That's it from my side. Thank you so much.